September's Target 2035 webinar. I'm Cheryl Aerosmith, and it's my pleasure to introduce today's moderator, Dr. Susanna Patanetseska from the Division of Neuroscience, National Institute of Aging at the NIH. Dr. Patanetseska is the director of the Office of Strategic Development and Partnerships at the NIH. She's developed a number of research portfolios and innovative programs in basic and translational research for Alzheimer's disease. She has a focus on building a systems biology and systems pharmacology capabilities for Alzheimer's disease research. And she also is developing a drug development program with an open science framework. This is an unusual and welcome approach to this area. So, and I think she's going to explore more of this in today's meeting. She's the program director for Accelerating Medicines Partnerships for Alzheimer's Disease, abbreviated as AMP-AD, and Target Discovery Program, and several AMP-AD affiliated open science programs and consortia. So Dr. Patanetseska, please take it away.
Thank you. Thank you, Cheryl, the organizing committee for the Target 2035 webinar series, Christina, Claudia, Ursula, and Target 2035 community. It's a real pleasure to be here with you today on World Alzheimer's Day and to share how our institute is using open science practices to enable the discovery, characterization of the next generation of therapeutic targets for Alzheimer's and to accelerate their entry into drug discovery campaigns. We have three terrific speakers who will highlight the work of the AMP-AD program and the TREAT-AD translational centers. So I just want to provide some housekeeping reminders. We look forward to engaging with you during post the presentations. So please remember that this webinar is recorded. Also, please make sure to put in your questions into the Q&A box. And the organizers have provided a short survey. So please take a couple of minutes to respond to it so that this webinar series can be even better moving forward. And also, before we begin, just a heads up to save the date for the next monthly webinar in October on the topic of open science antibody tools. There is a new date, but same time, same place. So now, without further ado, it is a real pleasure to introduce Anna Greenwood as our first speaker. Anna is a director at Sage Bionetworks and a leading investigator of the AMP-AD Data Coordinating Center and the Bioinformatics Core of the TREAT-AD Center at Emory, Sage Bionetworks, and the SGC. So Anna, welcome.
Thank you so much for the introduction, Susanna, and thanks to the organizers for inviting me today. So I am going to, as Susanna mentioned, I'm here representing both the Accelerating Medicines Partnership in Alzheimer's Disease and one of our two TREAT-AD centers, which you'll hear a little bit more about today. And I work at Sage Bionetworks in Seattle, so it's a little bit early in the morning for me still, but the caffeine is finally hitting my veins. So here we go. So Alzheimer's disease represents a major public health burden. More than 55 million people worldwide are estimated to suffer from AD or related dementias. But unfortunately, Alzheimer's drug development has been marked by repeated failures. In fact, there's been more than a 99% failure rate of clinical trials for Alzheimer's disease. Recently, you may have heard that aducanumab was the first AD treatment to be approved by the US Food and Drug Administration in more than 18 years. But this drug has been associated with significant debate about its clinical benefit. And moreover, it has a high price. And this really underscores a need for alternative strategies, even if the clinical benefit becomes more clear. It's thought that one of the contributing factors for this repeated failure rate is that for several decades, a lot of the drug development efforts for AD are focused on a small handful of therapeutic hypotheses, particularly around reducing the deposition of beta amyloid, which is associated with the neuritic plaques that are a hallmark of Alzheimer's disease. However, emerging evidence has demonstrated that the AD etiology is a lot more complex and multifactorial than just one single therapeutic hypothesis. And one line of evidence for this complexity has come from genetic studies, in particular, genome-wide association studies of large numbers of individuals with Alzheimer's disease. These studies have identified genes across several dozen loci that individually have very small effects on the overall risk of getting Alzheimer's. In addition, these genes are shown to be part of numerous biological pathways and are thought to affect the function of many different cell types in the brain. A second line of evidence suggests that the presentation of Alzheimer's disease varies quite significantly between individuals, and that Alzheimer's may actually consist of multiple subtypes. This is shown using several different phenotypic measures. I show two examples here, including imaging and biomarkers, as well as molecular signatures of gene expression signatures in the brain. So together, this evidence really suggests that treating any individual will likely require multiple complementary drugs that are targeted at different aspects of the pathophysiology of Alzheimer's. So we not only need new therapeutics, but this evidence is also telling us that we need a deeper understanding of the underlying disease biology. And so to help facilitate these efforts, the National Institute on Aging, which is part of the NIH, has several initiatives that are aimed at diversifying and accelerating the therapeutic pipeline. And Susanna Petencheska and her colleagues at the NIA have really led these efforts. So the first step of this pipeline are several NIA-funded research programs that are generating insights into novel disease biology and also identifying putative new Alzheimer's disease targets. Some of these researchers are also performing early validation on selected targets. These programs then feed their data and insights into several other downstream consortia that are generating resources to help accelerate the study of these targets. These include the Model AD centers, which are building next generation animal models for Alzheimer's disease, and the Treat AD consortia, which you'll hear a lot more about today. And importantly, and really championed by Susanna and her colleagues, all of these programs embrace open science principles and share all data and outputs with the broader research community without restriction. So at Sage BioNetworks, where I work, we serve as the data coordination center for several of these consortia, and we have built openly accessible infrastructure to distribute data, tools, and other resources that are emerging from these research programs. These include the AD Knowledge Portal, which allows exploration of data and tools coming out of the programs, and Agora, which is a results explorer that provides evidence around individual AD targets, as well as distributing a list of the targets that are being identified by these NIA-funded research teams. So how do these research teams go about identifying new AD targets? I'm going to describe how this is done by one of these research consortia, the Accelerating Medicines Partnership, or AMP-AD Consortium. So this figure just shows a high-level overview of the generalized process that AMP-AD teams are using. So the teams analyze tissue from human subjects from several different cohort studies that have both longitudinal and cross-sectional study designs. And then they use deep molecular profiling approaches, including genetics, gene and protein expression, and metabolomics and lipidomics. And these measures come from brain, blood, and or cerebrospinal fluid from these individuals. They then apply unbiased network modeling approaches to explore how gene and protein networks shift their activity as a function of disease. And then they extract the so-called network hub genes or key driver genes that are computationally predicted to actually cause these shifts in network function across disease. And then all of the data, insights, outputs of their network analyses, as well as a list of these prioritized driver genes or which are putative Alzheimer's targets, are shared with the broader community through the AD Knowledge Portal and the Agora platform. Of course, this diagram is overly simplistic. And if we look a little deeper, each team is using a different but complementary approach to identify targets. So some teams, like the team at Emory, focuses on protein expression. Duke University focuses on metabolomics and lipidomics. And all of them are nominating up to 100 targets and sharing them with the community. And so when aggregated together, these different methods and approaches really provide a rich list of new Alzheimer's disease targets for the community to explore. So these targets have all been shared through the Agora platform and can be accessed on the nominated targets page. Agora also shares targets that are nominated by external researchers outside of the AMP-AD consortium. And those can be, anyone can in the community can nominate a target by clicking this button and filling out a form, and assuming they agree to share their data, we'll add it to Agora. And the targets are annotated in this table with key information about how they were derived and other important information. So if we visit a gene detail page, we can see information about why each team nominated this target, including a description of methods and results, and a link to the raw data, if we click this button, that is housed in the AD Knowledge Portal. So these two platforms really interconnect with each other. In addition to details about why a target was nominated that's submitted by individual teams, Agora also shares unbiased molecular evidence, so you can evaluate the evidence yourself. These results are accessed through the evidence tab that's found on individual gene pages. And here I just show a summary of these different visual explorers that you can see. So there is differential gene expression and protein expression as a function of disease. So this plot is showing that in the parahippocampal gyrus, this particular gene is expressed at lower levels in Alzheimer's cases. And we see a similar pattern in protein expression. There's also data on metabolites as a function of disease. And there's a network plot that shows genes that are highly correlated in expression to the gene of interest. We also show correlations of gene expression or gene expression levels and some of the hallmarks of Alzheimer's disease, including pathological changes in the brain and cognition. So the AMP-AD teams have also been performing a variety of validation studies on some of their selected targets. And with validation studies, it's difficult to often to find these data because they're tucked away in a figure of a larger publication. And so to make these results more accessible, we have recently introduced experimental validation reports into the Agora framework. So these are descriptive reports from each team describing what the hypothesis that they tested was, what their model system was, reporting any statistical results, and then any links to publications if they exist. Agora also shares other information about targets, including an assessment of tractability. This was performed by scientists at Eli Lilly, who are actually part of the Accelerating Medicines Partnership collaboration. And this was published recently through TractaViewer. And so these results are accessible on Agora. We also provide links out to many other resources that I'm sure this audience is familiar with, including general target information resources, as well as disease-specific evidence sites. So a lot of the targets that are coming out of these systems biology studies are very novel, dark targets that very little is understood about. And so in order to de-risk these targets, they're going to need to be subjected to validation studies. But in many cases, even basic reagents are not available for these targets. So the Treat AD centers are focused on developing reagents for promising understudied targets and sharing all the outputs of the center's work openly and as rapidly as possible. So the TREAT AD centers are developing and sharing two main types of resources. These include insights about targets from bioinformatics analyses, as well as target enabling packages and chemical probes. And these are modeled after the work of the Structural Genomics Consortium. So two of TREAT AD's main bioinformatic efforts are around scoring and categorizing targets. So in Agora today, there are 609 unique nominated targets. And they come from such different analyses that it's difficult to do an apples to apples comparison to say which ones have the strongest evidence. So how do we evaluate these across teams? The TREAT AD center that I work at Emory Sage and SGC is working on a score to compare evidence across teams. In addition, these 609 individual targets don't represent 609 unique biological functions. So how do they collapse into related pathways? So I'm going to describe our center's approaches to address these challenges. So first, the TREAT AD Center at Emory Sage and SGC has developed a target risk score. This score quantifies the evidence for a particular target's association with AD across several different categories. This includes genetic evidence, evidence from different omics levels, including gene expression and protein expression data, also association from the literature, and a score of neuropathological association. So using these scores, targets can be more directly compared to each other to see which has the strongest evidence within these different areas. So these scores are going to be distributed through Agora. This is a design wireframe. It's not real data. All of these scores look the same here, but just to show how we're going to represent this data, it's being developed right now, and it's going to be available in Agora in a few months. So another effort of the Emory Sage SGC TREAT AD Center is to understand how these diverse targets collapse into shared biology and how they interact with each other in biological pathways. So our bioinformatics core has defined 14 biological domains that are based on several trusted sources and that represent the dysregulation associated with Alzheimer's disease. Gene ontology or GO terms are then defined for these biodomains, and then targets are assigned to these different biodomains. So we are working on finalizing these biodomain associations, and we will be sharing these on Agora as well. So in addition to the bioinformatics work, the TREAT AD centers are also developing target enabling packages and probes for a subset of the understudied targets. But how do the teams select which targets to develop these resources for? So I've been talking a lot about the Emory Sage and SGC Center, which I'm part of, but you'll also hear from Tim Richardson from the TREAT AD Center located at Indiana University and Purdue University. And each of these two centers is taking a different and complementary approach to target selection. So the TREAT AD Center at IU and Purdue is focused on developing a set of resources for targets within microglial cells, whereas the TREAT AD Center at Emory, Sage, and SGC is focused on de-risking understudied targets across many different dysregulated biodomains. But both centers are focusing on producing target enabling resources, which you'll hear more about in the next few talks. These resources are bundled into target enabling packages, or TEPs or TEPs. A basic TEP consists of a knockout cell line, a list of well-validated antibodies and purified protein. A full TEP contains additional resources, and in some targets are selected for chemical or biological probe development or even lead compounds. So these TEPs and component assets are being distributed within the AD Knowledge Portal. So if you visit the AD Knowledge Portal and go to the Explore Experimental Tools section and select the TREAT AD program, you can view the currently available resources that are already out there. New resources are being listed as soon as they meet a set of predefined quality control criteria. So check back frequently because this will be updated pretty much monthly. And so in conclusion, I just wanted to highlight where you can find the outputs of the Treat AD centers. So these will live in 2 different places, and we'll be linking between these platforms to make it so you don't have to memorize which place to go, but they'll be distributed through the AD Knowledge Portal as well as Agora. And so with that, I just wanted to highlight the huge effort of a lot of people that are part of the Treat AD Center, and to thank our champions at the NIA, including Susanna Petcheska and Larry Ruffolo, and the Treat AD Center at IU and Purdue, who are great partners as well. So thanks very much.
Thank you, Anna, for this terrific presentation. So this talk is open for questions. So please, again, share your questions to the Q&A function, but also the other panelists can just chime in through their audio with questions for Anna. If I may pose the first question, Anna. So the Treat AD centers have been ramping up their TEP operation throughout this long COVID season. Can you share with us what is the time horizon for the first wave of the release of the first wave of tools to the community?
Yes, sure. So our first target enabling package is available now. It's for a target called Moesin, and we have, I think, 6 or 7 antibody characterization reports that are performed by Icaros. And those are available now. And we have a large bolus of additional basic tests that will be available in the next few months, I believe. So.
Okay, so yes, please check the knowledge portal and Agora frequently. All right, we have, okay, I think everybody is warming up. The Q&A queue is being warmed up. Again, this is on a continuous stream, so please submit your questions as we go through the presentations. Are there any questions from the panel? And if not, we are then ready for speaker number 2. I'm delighted to welcome Tim Richardson, Scientific Director of the Molecular Innovation for Molecular Innovation at the Indiana Biosciences Research Institute, and he leads the Medicinal Chemistry and Chemical Biology core of the Treat AD Center at Indiana University and Purdue. So Tim, the floor is all yours.
Great, I share my screen. You should be seeing a starry background.
That's right, it's all stars, yep, beautiful.
Great. So slideshow from beginning, okay. And then got my laser pointer, okay. All right, so as Susanna described, I'll be representing the IUSM Purdue Treat AD Center. I'm a chemist by training, so I'll be from the medicinal chemistry perspective. The Treat AD centers, as Anna described, were established to create a new portfolio of biological targets for the treatment of Alzheimer's. We go through a bioinformatic analysis of data sets that are available to the community, as Anna described, and then we prioritize targets for lead discovery. The two centers do have slightly different focuses. The other center is focused more on dark targets. Our center is focused more on, I guess I would describe them more traditionally druggable targets, ones that we can move quickly into animal models and then have a trajectory to testing those hypotheses within the clinic. So we do have a goal of identifying not only probe molecules, but lead molecules. These would be molecules that have potency and selectivity for the intended target, and also good enough physiochemical characterization and ADME characterization such that they would have sufficient target engagement in animal models. And you'll see our center is collaborating very closely with the Model AD program. This is an initiative to create mouse models based on the bioinformatic analyses of human data sets. And we'll be working out PK/PD relationships within those mouse models. And then also, as Anna described, we will be putting together these target enablement packages and releasing the information all the way from in vitro biochemical assays to PK/PD measurements in vivo in those target engagement packages. Both centers are relying on the other NIA-funded initiatives, particularly AMP-AD, with respect to, as a chemist, I like to think about it as a molecular description of Alzheimer's. It's a deep mechanistic description of Alzheimer's based on those data sets, the genomic background of these patients, the transcriptomics, proteomics, metabolomics, correlated with the imaging and the cognition measures that define these patients as Alzheimer's patients. So that forms the basis of our disease target hypotheses. And as I mentioned, we collaborate closely with Model AD to test those hypotheses and mouse models that have also been built on this molecular description of human Alzheimer's. I did want to go ahead and put my acknowledgement slide up front. I think anyone involved in drug discovery knows it takes a fairly large team because of the many different expertise that are involved, everywhere from in vitro biochemistry all the way to animal models, PK modeling, and then PK/PD and everything in between. We do have a center that is organized along specific technical cores, a bioinformatics group, assay development, high throughput screening for the pharmacology, structure biology, and then the medchem group that I represent. Every good drug discovery team I've ever worked on has always begun with the end in mind. So I did want to have one slide in here about what our ultimate goal is, and that is patients and their caregivers and their physicians who want a treatment that can stop cognitive decline. And that cognitive function, there are a number of psychometric testing. These are scales that are used in the clinic to assess cognitive decline over time. And our goal is to halt that cognitive decline or even reverse that cognitive decline by this molecular intervention, targeting these biological targets that we've prioritized for drug discovery. One thing that is unique about the Alzheimer's area is the ability to visualize by PET imaging what's actually going on in the target organ of these patients. You can image amyloid deposition and neurofibrillary tangles, which are hallmarks of the disease. You can also measure brain function, for example, metabolism. And this allows us to observe what's going on in the brains of patients and identify patients early on in the disease before cognitive decline is measurable by psychometric testing, and then also understand at a mechanistic level what's going on behind the A beta deposition, the neurofibrillary tangles. Right behind that is a lot of work going on in biomarker development. And this would be an advantage in terms of being able to measure in a wider group of patients, CSF-based biomarkers or plasma-based biomarkers, and then correlate these measures of disease to the imaging and then to the cognitive decline. And this is something that we're very interested in. You see our center is focused on microglia and the role that microglia activation plays in inducing cognitive decline in these patients. And our center is starting to think about direct measures of target engagement, so proximal biomarkers. Our first two targets are SHIP1 and PLC gamma 2. So we're starting to think about what are the immediate downstream effects of target engagement with respect to these two enzymes and the role that they play in microglia, and then correlating those measures of target engagement to other biomarkers that are beginning to be established in the field so that we can follow target engagement preclinically and then follow target engagement clinically as well, and then correlate that to what the patients really care about, which is cognitive decline. So this slide just tries to bring all of that together. Anna did a great job describing the data sets and the bioinformatic analyses that are used to identify human risk and protective variants. The Model AD program is building those protective and risk variants into mouse models. The Model AD program is aging those models, so they have the late onset Alzheimer's disease risk built in. We're aging them, and we're aging them under stress conditions. We can, for example, just acutely stimulate with LPS, but we're also aging these animals on a high-fat diet, which tends to induce an inflammatory state. And we're particularly interested in neuroinflammatory state as mediated by microglia. And so with that, we developed target engagement biomarkers so we can follow how our small molecules engage SHIP1 and PLC gamma 2, and then correlate that to cognition and CSF and plasma biomarkers. I don't have time to do justice to the target selection, so I'll just work off of what Anna already described. We started with AD-nominated target list. I have a couple of leading references here, one from our own bioinformatics group, and then the Emory-Sage SGC bioinformatics group as well. We also took advantage of the target score that Anna described. And then we analyzed that gene list. We annotated it to microglia because that's our selected target cell. And then we're particularly interested in phagocytosis as a phenotypic readout of microglia activation. So we filtered through phagocytosis gene ontology terms, and then we used IPA, ingenuity pathway analysis, to build a network here. Here's the list of genes that we use to build the network. I always think about this as a plate of spaghetti and meatballs that you got to consume and kind of figure out what's going on. Each of these shapes represents a different biological entity. And based on IPA, these are the proposed relationships between those entities. And then we evaluate it with respect to our hypothesis that microglia activation plays a role in AD. We evaluate each target in terms of structure biology, and then based on druggable targets and strategies and technologies to drug those, we prioritize targets for lead discovery. So this summarizes that in more of a pathway view as opposed to a network, plate of spaghetti and meatballs. We were being a little bit open to what the actual connections are here between these entities, so there are not a lot of arrows drawn here. But we do believe that IMPP5D, or SHIP1, which is a phosphatase that takes PIP3 and converts it to PIP2, is an important driver gene in terms of microglia's involvement in AD. And also there are protective variants that upregulate PLC gamma 2. So another hypothesis we have is a small molecule that will activate PLC gamma 2. You can see there are several kinases and another phosphatase also involved in the pathway that we'll be bringing along behind IMPP5D and PLC gamma 2. In the context of Target 2035, I think most people listening in are probably familiar with resources such as Open Targets, Kemble, and ProbeMiner that we used to evaluate the biological entities and molecular tool selection for those targets. I thought I'd also mention TargetDB, a recently published chemoinformatics resource that we took great advantage of. This is there's a GitHub here. You can download this program, enter a gene in. It will give you an Excel file. It will score the targets across a number of different parameters. It does a great job of bringing a lot of information into a nice Excel file that you can then work from. I also mentioned our own bioinformatics group is building what we call Alzheimer's Disease Explorer. It's in beta test mode. There's a handful of genes that you can search on. And what we're attempting to do is bring in the aspects of these chemoinformatic tools that were very helpful for us in terms of evaluating these targets within the context of Alzheimer's disease and make this resource available to the community as well. So now I'm into our specific targets, SHP1 and PLC gamma 2. I mentioned SHP1 is, or IMPP5D is the gene name. It's a protein, it's a phosphatidylinositol phosphatase. So I've got the phosphatidylinositol pathway up here. SHP1 is here. It takes PIP3 to PIP2, so it cleaves that phosphate. It's generally viewed as a brake on the system. So a SHP inhibitor would be like taking your foot off the brake, activating microglia to a certain extent. You can see it's a multi-domain protein. There's an SH2 domain. It does tend to homodimerase. We've been able to demonstrate that. It also has a pH2 domain and a C2 domain, which will locate the protein at the intracellular side of the membrane so it can operate on the phosphatidylinositol. I've done the protein with the phosphatase domain here and the C2 domain here. One challenge in terms of ligand discovery is what type of ligand would be best to engage the target. So we've taken an agnostic view, at least in terms of the phosphatase and C2 domain, and tried to bring forward a number of different types of molecules that will engage the orthosteric site and a couple of allosteric sites that we've identified. So we ran an HTS at just the phosphatase domain. We would assume those are orthosteric inhibitors. We've worked up a fragment-based screen, and you'll see we've focused on fragments that bind into these allosteric sites. And also in the spirit of Target 2035, we've also done an exhaustive analysis of every SHP1 and SHP2 ligand that has been reported in the literature. We will be releasing all this data in our target enablement package. One of the nice things about this is the data in the literature is spread out across several different research groups. We'll be bringing all that data together in the same assay context, the same enzymes, assays, and the same cell-based assays. So we are testing these compounds at the phosphatase domain and the phosphatase plus C2 domain. And you can see in our hands for this selection of ligands here, these literature molecules tend to be about double-digit micromolar inhibitor potency in our enzyme assay. Interestingly, I thought I'd show this data here. This molecule here tends to destabilize the protein. So this is differential scanning fluorimetry in the presence of compound. Tends to actually destabilize the C2 catalytic domain to the degree about 1 degree to about 4 degrees Celsius destabilization of that protein. Activators, on the other hand, like this molecule, tend to stabilize that PTase C2 domain to the tune of about 3 to 4 degrees Celsius. Interesting data to show in terms of target engagement. We've also analyzed this publicly available fragment-based screen. This came from the Structured Genomics Consortium. They took a crystal of the protein and incubated it with a fragment library and published about 100 crystal structures. We analyzed the density for the small molecules binding at various places in that protein and decided to focus on site 7, both because of the quality of the molecular density, the small molecular density that was observed in this part of the protein, but also the types of compounds and the shape of that particular pocket was attractive to us. These are the molecules that, this is some data that we got on the fragments that were published. They were inactive in our hands. But you can see here we were able to add a covalent warhead because we noted this molecule was in close vicinity to cysteine 505. And when we added that covalent warhead, we got some inhibitory potency at the phosphatase C2 domain. And I'm showing an additional molecule here with some elaboration. We're trying to keep the molecules fairly small and drug-like so they can have exposure and brain penetration. And so adding this, or changing the small group here up top, we were able to bring the potency down to 8 micromolar in our hands with some good selectivity for SHP1 over SHP2. Okay. And as I mentioned, we also ran a high throughput screen. This is a reporter-based screen. We used this phosphatidylinositol substrate, and then malachite green as the reporter. It gives a fluorescent signal when you cleave phosphate, and phosphate interacts with malachite green. We ran about 100,000 compounds. This is from the Purdue collection up at Purdue University. And then based on that data, we did a chemoinformatic analysis for drug-like properties and selected several hit series that we then either resynthesized or repurchased compounds. And these are measured data here from resynthesized and repurchased compounds. And we're continuing to elaborate this series as well, and moving this forward under the assumption that these are orthosteric inhibitors, because we only screen the phosphatase domain in the HTS screen. Okay. So I'm going to switch over to PLC gamma 2. This is a complex enzyme. I think it would be a challenge for any drug discovery team. It has a multi-domain regulatory domain that protects the catalytic core from interacting with the cell membrane. Once this regulatory domain moves out of the way, the catalytic core will bump up against the membrane and cleave PIP2 and release IP3 intracellularly, and leaves DAG in the membrane. There are a number of activating mutations that have been observed in cancer. These activate the protein several fold over its basal activity. There's also this protective variant that's been observed in Alzheimer's disease. This particular variant will activate the enzyme to the tune of about 30%. And so that's kind of our window in terms of our hypothesis, which is we are designing molecules. We've analyzed this regulatory domain. We've identified some binding pockets based on a structural analysis. And our hypothesis is we can design molecules that will bind in this regulatory domain and either facilitate phosphorylation or destabilize the regulatory unit such that it's the wild type enzyme is activated in a similar degree as the protective variant. So that's our hypothesis with PLC gamma 2. Here's a quick look at our structural analysis. We used a homology model, a Swiss model, which is actually based on the rat PLC gamma 1 structure, which has been published. Identified a couple of binding sites, particularly site 5, which we're interested in. Just like SHIP1, we have done an exhaustive evaluation of literature compounds. Unfortunately, most of the ligands that have been published for PLCs actually don't engage the target, except for ATP, which was recently published by John Sondek's lab. We've been able to confirm that observation, and some ATP mimics as well. And here's some of that data. Very similar data to what the Sondek lab published for PLC gamma 1. We've shown a KD of ATP of about 1.4 micromolar. We've also run differential scanning fluorimetry against PLC gamma 1 and 2 wild type, and risk variant and the protective variant. And we demonstrated that ATP engages all 4 of these enzymes. And then finally, we have an enzyme reporter assay that I'll show you and demonstrated that ATP inhibits PLC gamma 2. Sondek was able to show that it inhibits PLC gamma 1. So we've been able to confirm those results as well. So we believe we have a legitimate ligand now, ATP, that engages the target. We decided to move forward with an affinity selection strategy. Here we incubate the protein with actually mixtures of compounds using ATP as a control. And then there's a surface that the protein binds to. And then you measure by mass spec compounds coming off the surface. And then you can, obviously the molecules that get held back on the surface by the protein, those are ones that we believe are engaging the target. So here's a quick look at some of our data. So this is ATP as the control. Does seem to have a little bit better affinity for PLC gamma 1 than PLC gamma 2. Here's a molecule that hits both proteins. So this is the relative signal over background for both proteins. This is a molecule that appears to be selective for PLC gamma 1. And here's a molecule that's selective for PLC gamma 2. And these are showing you relative signal versus signal to background We have about 15 or so molecules, each representing a different scaffold that will confirm in IC50 and move forward. Okay. And I do want to mention real quickly, I'm bumping up against my time here. We do have a close collaboration with John Sondek and Chiseng Zhang. We're actually using their reporter assays. They have a solution-based assay and a liposomal-based assay, and we're evaluating the kinetics of activation using this reporter enzyme construct. Okay. And I'm out of time here, so I'll skip the conclusion slide, and I'll just end
Tim,
with
I think
my
we're doing
acknowledgements.
Tim, we're doing well on time, so you can please take a minute to cover your last slide. I think we're
Okay.
perfect.
Yeah, so to summarize here, we're building a portfolio of biological targets, assays, probe molecules, and leads. They'll be released in target enablement packages. And the goal here is to facilitate the discovery of new therapies for Alzheimer's. It is a team science effort, so I want to acknowledge all of my colleagues that have been involved in these projects. And you can see we've done this in an open science context. So, for example, the collaboration with John Sondek and Chiseng Zhang. So we definitely invite additional collaborators who might like to work with us. We can test compounds, we can send compounds out, particularly for SHIP1 and PLC gamma 2, which we have enabled. We completed a thorough review of the literature molecules. We'll be releasing all that data together and the target enablement packages. And eventually, I know there's a question about timing. In terms of timing, we'd like to have our TEP, our initial TEP for SHIP1 published by the end of the year. And then we'll work on a full TAP for SHIP next year and an initial TAP for PLC gamma 2 next year as well. Okay.
Thank you, Tim. This was fantastic. And so we have one question, which I think is both for you and Anna. I just wanted to share it with everyone from Dr. Madhav Tambisetti. Apart from generating TEPs, are there plans to acquire more downstream validation, such as phenotypic screening against targets? So Anna, if you don't mind sharing your response first, then we'll then go to Tim.
Sure, absolutely. So we are doing some early validation experiments for a limited set of targets. In particular, these are such dark targets that sometimes something is very promising, but we don't even know what direction we would want to move that target. So, you know, we do some basic work in iPSC cells lines to evaluate the directionality, but we're trying to establish collaborations with the experts who are already studying these targets, maybe in other diseases or other contexts. And so if that's you, please reach out to us. We'll be sharing our current portfolio, and if we'd love to collaborate.
Yeah, and I can describe, I didn't have time to put the cell-based data in here. What we have, we actually do have a phenotypic screen. So we're actually initially working with BV2 cells and HMC3 cells, which are immortalized mouse and human microglia, respectively. And we're measuring phagocytosis with photo row labeled myelin. And then we also measure cell number and nuclear area. And we bring forward compounds that upregulate phagocytosis and have minimal effect, particularly on nuclear intensity. If we see an increase in nuclear intensity and a decrease in cell number, that indicates that the compounds are cytotoxic. So that's a kind of a high-level description of our phenotypic cell-based assay. We'll also be measuring the same phenotype in mouse primary cells, and then yet to be worked out PK/PD relationships in the model AD mice.
Thanks, Tim. And there is a follow-up question for you, Tim. Do you have a prediction regarding what your desired phenotype is for agents that target SHIP1 and PLC gamma? Have you prioritized leads from HGS to assay in phenotypic screens?
Yeah, one thing I actually do like about our first two targets is biology's already kind of done the experiment for us, which I definitely appreciate. So this this, it's always good to have evolution do the drug discovery for you, right? So, this protective variant, it's as protective in Alzheimer's as ApoE4 is risk. And so we plan to use that protective variant. I could also show this slide here. So here's an example. There's a risk variant, M to L 28 variant, and a protective variant, P522R. And we do plan to follow those activities as a window of how much activation that we would need from the wild type to, say, a small molecule that would bring the wild type up to the same level as a PLC gamma P to 522R. So the way I think about it, that kind of gives us an efficacy window that we can work with in a phenotypic sense. And I also mentioned there are some activating mutants. We haven't done this yet, but we plan to make the activating mutants which activate PLC gamma 2 several fold and tend to have a risk for oncology. So I think that gives us an efficacy window with respect to a protective variant, and then a safety window with respect to what too much activation would look like in the cell type.
Thank you, Tim. So I think, again, please continue to with your questions as we move along. We will also have a segment, a little more time at the end for some more discussion. If there are no other immediate questions, we can move on. And it's my pleasure to introduce our last speaker today, Vittorio Katis, researcher at the Center of Medicine's Discovery at the University of Oxford, and a key member of the structural biology and assay development team for the Treat AD Center at Emory University, Sage Bionetworks, and SGC. So Vito, welcome.
Thank you, Susanna. Okay, as Anna has already mentioned, most therapeutics for Alzheimer's target either amyloid beta or tau. And these have shown very limited efficacy in clinical trials. And there is definitely a need to explore novel Alzheimer targets. So as part of Treat AD, our goal is to catalyze the diversity of Alzheimer target pipeline. And we want to do this in an open way in order to promote the study of understudied Alzheimer genes. And through this, we want to support the emerging Alzheimer therapeutic hypotheses and provide high quality reagents, such as drug-like compounds, to the scientific community. And we want to share everything without restriction. At Treat AD, Emory, Sage, and SGC, we currently have over 20 targets as part of our portfolio. And as Anna was mentioning, they're part of 14 biological domains. We want to do a basic TEP for all of these. And a basic TEP, as Anna was mentioning, includes purified proteins, knockout cell lines, and validated antibodies. And in a smaller subset of these, we want to also produce biochemical and biophysical assays, crystal structures of the proteins that are relevant, and Alzheimer relevant knockdown cell lines. And eventually we want to have hit compounds that hopefully will lead to a cellular probe. So in this talk today, I just want to focus on two of our current targets. And this is work that has only been in progress for a year or so. So it's quite preliminary. So in Alzheimer's disease, it is characterized by brain atrophy and the accumulation of amyloid beta plaques and tau neurofibrillary tangles. And innate immune cells that include microglia have shown to have important roles in the pathogenesis of Alzheimer's. In Alzheimer GWAS studies, rare genetic variants of inflammation genes that are expressed in microglia have been identified. Now, microglia can have beneficial roles in removing amyloid beta plaques, but it can also have detrimental roles. Hyperactive microglia can actually cause neuronal damage by the production of inflammatory cytokines. So for microglial protective functions, you want a balance of activities. As I mentioned before, therapeutics targeting amyloid beta and tau have largely failed in phase 3 trials. And there is now an increased focus on new targets involved in inflammation. And we have employed a multi-omics approach to identify new Alzheimer targets. As Anna was mentioning, we integrate large-scale molecular data that includes genomic, transcriptomic, epigenomic, and pathologic from human brain samples. And these are from Alzheimer patients or from normal subjects. And then with that data, we produce a network model in order to identify targets for experimental validation. And on the right side, you can see our prioritized targets mapped to various biological domains. And SIK and FCR1G are both involved in the innate immune response, but there are quite a few other targets that are also involved in the immune response. Both SIK and FCR1G are highly upregulated in microglia. This is a neuronal cell type enrichment analysis, and it forms part of this, what we call group 6. FCR1G is a subunit of the Fc receptor complex. Essentially, it has a very small extracellular region, a single transmembrane domain, and a cytoplasmic tail that contains an ITAM motif. And it forms part of 3 different complexes. It's a complex that binds IgE or IgG with low affinity or IgG with high affinity. So upon immune complex formation and binding to the Fc receptor, the ITAMs of FCR1G become in close proximity. And this results in phosphorylation of the ITAM motif by the Lin kinase. And this allows SIK kinase to be recruited. So the ITAM consensus, which FCR1G contains, is a tyrosine, which is phosphorylated, 2 residues, and then a hydrophobic residue leucine. Then there's a spacer region, followed by again a phosphorylated tyrosine, 2 residues, and then leucine. SIK kinase has at its end terminus 2 SH2 domains. And here I show a published structure of the full-length SIK kinase. The ITAM engages the SH2 domains, and this results in activation of the kinase by a mechanism which is not entirely clear, but one hypothesis is that the signal is transmitted through to this linker region, which moves away from the kinase and allows it to be phosphorylated and then activated. Now, what is the evidence for FcR1G and SIC being involved in Alzheimer's disease? The Fc gamma receptor 1 and 3 are both upregulated microglia in neurodegenerative diseases, such as Alzheimer's. The Fc gamma receptor 1 expression in microglia was reduced in Alzheimer patients that have been immunized with amyloid beta. Furthermore, FcR1G is a GWAS hit for Alzheimer's, and in mice APP models, it is highly upregulated in activated microglia that are associated with neuroinflammation. SIC is also significantly upregulated in Alzheimer brains. There was no GWAS association with SIC, but it is upstream of several other GWAS hits, such as CD33, and as Tim was mentioning, TREM2. Now, TREM2, TYRO-BP, or otherwise known as DAP12, does activate SIC, and it's this activation that has been implicated in Alzheimer's. So our therapeutic hypothesis is that preventing binding of SIC to FcR1G would reduce the detrimental inflammatory activity of microglia in Alzheimer patients. There are quite a number of potent SIC inhibitors that have been identified, but we wanted to focus on this protein-protein interaction. So in doing so, we produced the SH2 domains of SIC and managed to crystallize it in the presence of a phosphorylated ITAM of FcR1G as a peptide. And we can see, this is a novel structure at 2.2 angstrom, and we can see that the phosphotyrosines bind into this basic groove or pocket, and the hydrophobic residue, leucine, into an adjacent pocket. And this is sometimes called, a two-pronged plug into a socket mode of binding. And this is repeated on both of the SH2 domains. So we noticed that the N-terminal phosphotyrosine, the phosphate group engages 3 arginines in the pocket. In the C-terminal phosphotyrosine, we notice that the phosphate in some cases engages the 2 arginines and the lysine from the other SH2 domain. But in other cases, this arginine 45 shifts down and comes in close proximity to the phosphate groups. And this was somewhat curious because in the crystal we obtained, we had 6 different molecules, 3 of which was of one type and 3 of which was the other. And then when we looked at the backbone of the ITAM across the surface of SIC, we saw that within the central region between the 2 phosphotyrosines, that the backbone diverged. And this corresponded to the 2 different modes of binding of the phosphotyrosine. And this was curious, but then we also noticed that they correlated with the position of this linker region between the 2 SH2 domains. We also solved SIC in the absence of the ITAM, and when we compared it to the ITAM bound form, in one mode, this linker region superimposed, while in the other mode, there was a large displacement of this linker region up to 12 angstrom. Otherwise, the SH2 domains didn't move very much at all. So I'm proposing that perhaps in this mode 2, this is the activated form of of SYK. And this large movement of this hinge allows the kinase domain to become phosphorylated and activated. However, this is just speculation at the moment, and I have to confirm this by mutagenesis and activity assays. We also produced structures of the SH2 domains of SYK bound to 2 other ITAMs, including TYRO-BP, and all of the ITAMs bind in a very similar fashion. And this suggests that it might be difficult to selectively target the FcR1G SYK interaction over any other ITAM SYK interaction. So we measured the affinity of the ITAM to SYK by SPR, and it binds very tightly with a KD in the low nanomolar. We developed a TF-FRET-based assay, a protein-protein interaction assay, where the donor fluorophore is bound to a his-tagged SH2 domain of SYK, and the acceptor fluorophore, FITC, is on the FcR1G phosphor ITAM. So when they two bind, we get a FRET signal. And we validated this assay by titration of the signal with an unlabeled phosphor ITAM. And this was specific to the phosphorylated tyrosines, because when we had a non-phosphorylated peptide, this was unable to titrate the signal. We performed a pilot high throughput screening using the assay that I just described, but adapted for high throughput screening. And we used an FDA approved and bioactive compound library. And we found actually quite a number of hits in the low micromolar range. In particular, there was one hit, CCG 50014, that was in the nanomolar range. And then when we repurchased this compound and tested it, we found that it could inhibit the interaction of the ITAM with SYK in the low nanomolar. And this interaction was not specific to FcR1G because it inhibited the interaction of all the ITAMs that I tested. So we went ahead and performed the primary high throughput screen with the 138K chemical diversity library. And we got 294 hits, which represented a 0.2% hit rate. And of these, we confirmed 135 of them by dose response, and some examples are shown at the bottom. These are in the low micromolar range. 112 of them were confirmed with reordered compounds. And then we did, 112 were reordered and 54 of them were confirmed. And then we performed a counter screen using a TF-FRET assay, which is analogous to what we use, but with a different protein and peptide, and also a pains analysis. And we were left with 43 compounds that contained 13 different chemotypes. And that's as far as we got at the moment. We're currently performing a secondary screen using a GST pull down of full-length proteins in a cell lysate. And soon we'll begin evaluation for medicinal chemistry. So in summary, for SYK and FcR1G, we have produced protein and reagents, structures, both ligand bound and APO, and developed a primary TF-FRET-based assay. And at the moment, we have a number of hit compounds that we hope will soon go into medicinal chemistry for SAR, and then hopefully eventually probe development. At the moment, both SIC and FCR1G are undergoing target validation using microglial iPSC-derived cells with a CRISPR knockout, and also this inhibitory and active CRISPR. And we're also developing secondary cellular activity assays, which to look at SIC activation by clustering the Fc gamma receptor using monoclonal antibodies. And that's it. Thank you.
Thank you, Vito, for an excellent presentation, taking us on this really arduous, exciting journey. And so we are open for questions, and we have some time for a longer discussion.
Yeah,
So
I just wanted to, I forgot to go to my acknowledgement slide, of course. And I just wanted to thank our whole Treat AD Center at Emory Sage and SGC, the high throughput screening team, the admin. I'm part of the structural biology group, the medicinal chemists that we have, and of course the bioinformaticians. And I would like to thank NIH and NIA for funding Treat AD. Sorry, thank you.
No worries, thank you. Thank you, Vito. So we have several questions that have come through. So I will, I believe one was answered, but let me ask it again in open forum by Dr. Gregory Salvin to any and all the speakers. How do you see the release of AlphaFold human proteome guide your future AD target selection? And Tim, maybe start with you, even though I think you posted your comment.
Yeah, we would use AlphaFold similar to other homology models. I think one nice thing about AlphaFold is it does bring everything together in one whole view of the protein. So that is one advantage of AlphaFold over some traditional homology models. It does give you a starting point for placing various domains in three-dimensional space.
Yeah, I would like to add that AlphaFold is actually very useful to guide us in designing constructs for expression of protein, because it can tell you the domain boundaries of the different domains within a protein, and that can really help to make soluble protein, which can sometimes be difficult.
It's a good time saver.
Yeah.
Thank you both. So then we have a question from Dr. Takis Prinos. There are clinical SIC inhibitors for autoimmune diseases and oncology indications. Wouldn't these be more accessible and straightforward tools to validate SIC as an, well, it says HD target, but I'm assuming AD target. Have you tried any of these in your Treat AD models?
Yes, well, we are currently establishing our cellular models for Alzheimer's, and we have put together what we call an informer set. This is also one of our deliverables, where we take all of these compounds and put them into a panel where people can get them from us, or later when we've fully developed our cellular models, we'll test them ourselves. So we're trying to set up, for example, models with iPSC microglia to look at phagocytosis, among other things. And so definitely we will look at the already available SIC inhibitors. But we want to contribute something novel. I mean, they're already out there. The purpose is that we want to try to inhibit the SIC in other ways, maybe more specific to what we want.
Thank you.
I could just add that there's a preprint out there on that AD informer set. There are, I believe, 4 SIC inhibitors in that set. We've tested them in our cell-based assays. They do have activity. They're not super compared to some of the other molecules in that set. Another thing I'll point out is, most of those molecules weren't designed for central target engagement. So there's some aspects of brain penetration that would need to be optimized for those molecules in an Alzheimer's context.
Thank you, Tim. So as we wait for questions, and again from the panelist gallery or the audience, let me ask you a question actually to all of you. As you know, so this massive molecular redefining of AD with data and this molecular mapping of the disease that is happening across the AD research field now is, of course, going to help us take a more precision medicine tailored approach to drug discovery and drug development. And so as you are making the selections and prioritizing the targets for which you are either developing tools or taking them into drug discovery, how are you bringing these precision medicine principles into those considerations? Targets suitable for early disease or for more restorative therapy or for a subtype, or is that part of your iterative process? Decision making on what to choose and how to have the end in sight. So
well, I can speak a little bit to that. So within our center, we are one of the ways we're evaluating these targets is which stage of disease they're predicted to act in. So are they earlier targets? Are they later targets? And we're using a lineage tracing approach to figure out which genes are active at early versus late stages. And so we are trying to select a diverse portfolio where some of those targets in our set are earlier and some are expected to be later. So that's just obviously one piece of this larger puzzle.
yeah.
And I'll just add that's a particular challenge for the targets that we've selected, where SHIP1, PLC gamma 2 would be that target engagement we would hypothesize would be upregulate microglia. So I think a good question is, at what stage in the disease would you really want to upregulate microglia? So we're anticipating that the world of biomarkers and Alzheimer's will advance to the point where we can stage patients and we can connect our target engagement biomarkers to biomarkers of inflammatory states that have been correlated with clinical progression, and then try to stage treatment to the patients that would benefit most from upregulating microglia.
Okay, so looking at the Q&A queue, and we have a somewhat shy audience today, but there's no bad questions, so please feel free to query our speakers. And so as we're giving folks a couple more minutes, just to the speakers, and so particularly Anna, Tim, as you know, the NIA is supporting a large community of drug discoverers, drug developers through our other funding opportunities, and we're producing their whole array of new INDs for a diverse portfolio of targets. And these have been so far, developed through more traditional methods. We'd like to think that your work is going to be enabling not just of new drug discovery, but also helping those who have delivered clinical drug candidates maybe use the data, knowledge, and tools to further drive the clinical drug development in a more data-driven way, or learn more about the biological domain they're targeting. How do you see the value of your program to actually, for that community of drug discoverers who are already in the process of preclinical drug development and delivering clinical leads?
I think some of the bioinformatics resources that we're developing and sharing through the Agora platform will be very useful to that group of researchers. In particular, I didn't go into detail, but the biological domains that we're classifying targets into is just the first start. And then we're actually also aggregating groups of targets into therapeutic hypotheses. And so once we do that and assemble these therapeutic hypotheses and share those, hopefully some of these other, people who are working on a particular target can kind of come and see what other targets it's really thought to be involved in. And so obviously you can do that from the literature now, but we're sort of applying new novel approaches
Principles, yeah.
and principles to do that. Yeah.
I think of the target enablement packages as hit declaration packages, so as scaffolds that have been well characterized, and a flow scheme, biochemical assays, cell-based assays, and in vivo models that can be used to optimize those molecules. And then the next stage would be more lead development, where you bring in not only the ADME characteristics and PK/PD, but also the toxicology, which would give you a foundation for an IND filing for the FDA. So I think about the target enablement packages as like hit declaration, and the next step would be lead development.
Thank you. All right, so we do have the reminders to fill out the survey, folks. I just want to take a minute to give an opportunity, go back to our host, Christina, Ursula, for any last minute announcements or messages to our community before we adjourn.
Thank you so much, Susanna. Thank you to all the speakers and today's moderator and host. Stay tuned for the next webinar, which is upcoming in October. We also have an interesting one on solute carriers in November. So check out the information on the target2035.net website.
Thank you, Christina. I just want to thank everybody again. Our speakers are the Target 2035 organizing committee. And I hope that we were able to show the value and the power of open science in early drug discovery and really gaining deeper understanding of the disease biology, which is really central to being successful downstream. And as they say on the airlines, we know that you have many webinars to choose from, a wealth of exciting stuff to look at every day, all day. So thank you for choosing to spend this hour and a half with us. And we wish much success to the Target 2035 initiative and to growing this community. Thank you all.